Good morning, everyone. My name is Slżbieta Skorupska and I am professor at the Poznań University of Medical Sciences. Today, I would like to present the complex results of my research team's work on the objective diagnosis of referred pain. Let me begin by outlining the agenda for today's presentation. First, I will briefly explain what referred pain is, highlighting its clinical relevance and diagnostic challenges. Next, I will discuss the role of thermography in detecting referred pain patterns. Then, I will move on to electronography, showing how it can help identify the motor components of referred pain. After that, I will present the outcomes of our research and in connection with the central sensitization. Finally, I will conclude with a few points on the clinical implication and how our findings can be translated into practice. In the field of somatic pain, most clinicians are familiar with radicular and neuropathic pain, but much less is known about referred pain. Referred pain occurs in the area distant from primary lesion and is sometimes associated with secondary hyperalgesia and tropic changes in the affected region. Although clinically common, the nature of referred pain remains poorly understood. In many cases, pain occurring in dermatomal regions innervated by specific peripheral nerves is simply classified as radicular or neuropathic pain. Addressing this issue, the International Association for Study of Pain, citing a classic treatise on pain, started back in 2011 that pain in the lower limb should be described specifically as either referred pain or radicular pain. This recommendation should also be applied when diagnosing pain in other somatic regions. Somatic pain may originate not only from pathologies of local tissue, but also from dysfunction in distant structures, including the spine, neuromuscular system, and other somatic components. The term referred pain is used to describe pain perceived in somatic areas, far from the site of noxious stimulation, which is not caused by nerve irritation. However, it is difficult to accurately map referred pain areas, as referred pain from different origins often overlaps. Moreover, referred pain does not follow dermatomes or the distribution patterns of nerve roots. It is clear that referred pain is both common and variable. For example, spinal referred pain reported occurs in 70 to 84% of patients with low back pain. As early as 1939, Kellgren developed an experimental model demonstrating that referred pain could be triggered by the injection of hypertonic saline solution, and a finding later verified in cases of spontaneous pain with unclear etiology. Building on this concept, various percutaneous procedures such as provocative lumbar discography, facet joint injection, nerve blocks, intra-articular sacroiliac joint injection, and epidural injection have been developed to reproduce or block referred pain and to identify its exact source. Inflammatory pain models, capsaicin has also been used in the functional spinal and supraspinal magnetic resonance studies, which have been reported to help localize the origin of referred pain. To confirm that pain reported by the patient is referred pain, two main diagnostic strategies can be used. First, we can try to reproduce the same pain pattern by applying different stimuli, for example, mechanical, thermal, or chemical. If the patient reports a similar sensation in the same remote area, this supports the diagnosis of referred pain. The second approach is to relieve the pain using a local anesthetic block. If the referred pain subsides after the injection and the suspected source, this also confirms its origin. These two methods are often used together in both research and clinical practice to increase diagnostic accuracy. But please remind that this is a solution for joint referred pain, not for trigger points. My petted solution allow us to visualize a specific physiological response to trigger point stimulation, the so-called referred sensation. What makes it unique is that it doesn't reflect pain itself, but rather the autonomic component of referred sensation captured in real time during dry needling. This innovation led to a patent application and received multiple international awards, confirming its scientific and clinical relevance. Thanks to that, it opens the door to objective imaging of trigger point related response, which may support better diagnosis and understanding of myofascial pain. In our research, we use thermography to measure not only the temperature but also the size of the area with increased temperature, which can correspond to the referred pain patterns. What you see here is a representation of the concept of isothermal areas, that is, regions with the same temperature. Each image shows the isothermal area for a different temperature range. By analyzing how these areas expand or shift, we can objectively identify and quantify the thermal expression of referred pain. Our diagnostic protocol undergoes a process of validation and reliability, which is essential for ensuring its effectiveness and reliability. The muscle examination is based on the noxious stimulation, 10 minutes of fast in, fast out dry needling, followed by 6 minutes of further observation of the patient at rest under infrared thermal camera control to detect the expected vasomotor response coincident with the referred pain zone in the examined muscle. The two SP test phases, stimulation 10 minutes and observation 6 minutes, are consistent with active dynamic thermography protocol demands. Cutoff temperature is determined on the initial thermal image before the SP test starts and is defined as a temperature at which the smallest isothermal area with the highest temperature on the thigh, calf, or foot is visible. The MATLAB implementation resulted in lots of new observation. Firstly, we can observe the development of the size of the phenomenon and the average temperature increase every 3 seconds. Then we saw that the minimum time for stimulation is 4 minutes. Additionally, MATLAB allows us to measure the statistical significance of each single record from the thermograms every 3 seconds. The results of the statistical analysis can be seen in the bottom right-hand pictures on the slide over here. The final stage of the protocol development involves defining the vasomotor phenomenon due to dry needling stimulation as a one parameter. After that, we confirmed amplified vasomotor reactivity based on the percentage site of abnormal autonomic activity coincident with reported referred pain during stimulation. As a part of further development of our diagnostic approach, we integrated the infrared thermography based system with electromyography. Here we focus on motor phenomena associated with referred pain. Specifically, we use the SP test, which provoke a pain response, and combined with EMG recording to assess muscle activity patterns. To analyze this, we use both a common superficial EMG signal with the short-time Fourier transformation and the NeuroMap software by Delsis, which allow us to automatically extract and visualize motor unit shapes and behaviors. Unfortunately, we haven't analyzed the outcomes of NeuroMap yet. We investigated the motor responses in the thigh area. Electrodes were placed on the 8 muscles, the gluteus maximus, gluteus medius, semitendinosus, biceps femoris, vastus lateralis, vastus medialis, rectus femoris, and adductor longus. We then performed 10 minutes of noxious stimulation to the gluteus minimus using a thin filiform needle. So what were our changes? We observed that in most participants, when referred pain appeared, a specific and selective bioelectrical response also occurred. Initially, we hypothesized that we could detect changes in the superficial EMG signal by comparing bioelectrical activity during a maximum voluntary contraction with the signal recorded during the SP test. However, normalization failed, and we had to take a different approach. To overcome the challenges with signal normalization, our bioengineering collaborator proposed to use the short-time Fourier transformation analysis. This method allowed us to examine the frequency content of EMG signal over time. The spectrograms presented here display the power spectral density, which help us visualize how muscle activity evolves during the SP test. While we won't go into the technical details now, what is important is that we observe a notable increase in muscle activity precisely in the area where referred pain was reported. This increase was confirmed not only by the patient's subjective experience, but also by the objective findings from the SP test. The slide presents the results of a short-time Fourier transform analysis comparing muscle bioelectrical activity between individuals with referred pain, experimental group, and those without it, a control group. The features on the left visually illustrate differences in spectral power distribution across various muscles. The table on the right summarizes the median power spectral density value with the muscle showing significantly higher activity in the experimental group highlighted in the bold. Statistically significant differences were observed in the semitendinosus, vastus lateralis, rectus femoris, adductor longus, and vastus medialis, indicated an atypical motor response associated with referred pain. We have already published our first work that present the results of our short-time Fourier analysis, which you have seen on a previous slide. The concept of the link between trigger points and nociplastic pain is not my own. The World Health Organization group of the experts working on defining chronic pain as a disease consider the trigger point concept necessary for defining cervical or radicular spine pain syndrome for primary chronic musculoskeletal conditions. Finally, they removed trigger points description because they posited that the need of the more empirical data. Our research led us to a crucial question. Is referred pain purely a nociceptive sensation, or does it reflect a form of nociplastic pain? Nociplastic pain is characterized by altered nociception without clear evidence of tissue damage or nerve injury. It often involves not only sensory disturbances but also autonomic and motor symptoms, which are increasingly observed in clinical settings. As shown in the diagram, trigger points described by Travel and Simons can produce referred pain patterns involving all three domains: sensory, autonomic, and motor. Taking together these findings suggest that referred pain, especially when persistent, may not be purely nociceptive in nature, and some subtypes of the nociplastic mechanism can be considered. The first condition of nociplastic pain concept is confirmation that abnormal autonomic activity is involved in the development of pain. In our studies, we confirmed that a higher proportion of cases presented with amplified vasodilatation, which can be defined as abnormal autonomic activity, exclusively characterized trigger points. Additionally, we confirmed that in a few cases, amplified vasoconstriction was present. For more information on the potential connection between trigger points and nociplastic pain, please refer to the studies we have published. The future studies should not only focus on a back pain, whether accompanied by leg or arm pain. Clinically speaking, chronic primary pain is also known as functional pain. As we know, all musculoskeletal problems with referred pain are currently categorized as functional pain by clinicians. Referred sensation were recognized as an interesting topic in sport medicine. Athletes are more likely to develop more trigger points than people who lead sedentary lifestyle, according to some studies. Latent trigger points are often clinically silent but can still impair muscle coordination, motor control, and even contribution to referred pain during overload, fatigue, or stress. In athletes, such dysfunction may go unnoticed until they lead to decreased performance or injury. Using trigger point concept and diagnosis of latent trigger points can support both early intervention and individualized rehabilitation plans, offering a more preventive and more objective strategy for managing pain in elite sport context. Based on the data from preliminary version of the 11th revision of the International Classification of Disease, further studies should focus on back pain and low back pain and leg pain. Additionally, trigger points coincidence with other musculoskeletal disease or fibromyalgia and other diseases. Forming the list of chronic primary pain are recommended. A few take-home messages for you. Referred pain from trigger points, especially when not linked to visible structures damage, may actually reflect a nociplastic mechanism, and that changes how we should think about diagnosis and treatment. Our research showed that thermography and electromyography can offer objective evidence of these processes, even in cases that are often dismissed as non-specific pain. This clear clinical implication, it supports early identification, enables target interventions, and opens the door to prevention strategies, particularly important in musculoskeletal medicine and athlete care. Thank you for your attention. If you want to contact me, please write an email.